Okay, so welcome back to this next video in which we are discussing acid-sensing ion channels. Okay, right, so we have discussed that acid-sensing ion channels, also called ASICs, are members of the P2X-like receptor family of ligand-gated ion channels, which means that their overall structure is a trimer made up of 3 separate subunits joined together, okay, to make the full ion channel. And each of these individual subunits has 2 membrane-spanning alpha helices, with the amino terminus and carboxylic acid terminus both on the cytoplasmic side, and therefore a loop between the 2 membrane-spanning alpha helices on the extracellular side. Okay, right, so let's just copy that information down onto this side of the piece of paper. So, overall, the ASIC ion channel looks like so. Okay, so you have this trimer of 3 separate proteins joined together to make the entire ion channel like so. Okay, then if you take a single one of the subunits out and have a look at its membrane spanning topology, what it looks like is so. You have the amino and carboxylic acid termini on the cytoplasmic side, and then these 2 membrane-spanning alpha helices with a larger loop on the extracellular side between those 2 membrane-spanning alpha helices. Now, the incredibly simple thing would be if there was just a single subunit called the acid-sensing ion channel subunit. Okay, you use that gene 3 times, made 3 proteins, joined those proteins together to make an entire acid-sensing ion channel, and that was it. Okay, it's not that simple, however. There are multiple genes for acid-sensing ion channel subunits, and they have multiple splice variants. Okay, so at present there are 4 known genes for the acid-sensing ion channels, and this is where I really did get the information from my reference, Kellenberg and Grutter, 2014. Okay, so there are 4 genes, okay, called the ASIC gene 1, okay, which I'll just denote ASIC 1, then the ASIC 2 gene, okay, like so. Then we have ASIC 3 and also ASIC 4. Okay, so that bit's very nice and simple. However, there are 8 known different subunits for ASIC channels. Now this looks like a bit of an anomaly, because how can we have 4 genes and yet 8 different subunits? How can 4 different genes produce 8 different subunits? Okay, so we need to now talk about the concept of different splice variants. Okay, and for this we're going to revise the central dogma of biology. Okay, so let me remind you of the central dogma of biology. So, let's say this is the double-stranded DNA. So we've got 1 strand here and the complementary strand of DNA here. And let's say we have a gene for, well, let's say this is ASIC 1. Okay, so this is the gene for ASIC 1. Now, basically, only one of those 2 strands will actually be used by the RNA polymerase enzyme in order to produce a strand of mRNA from. Okay? So the strand which the RNA polymerase actually works along and produces a piece of mRNA which is complementary to is known as the coding strand. So this strand that I've coloured in turquoise, let's suppose that this is the coding strand of the gene. Okay, and what will happen is the RNA polymerase enzyme will work along this coding strand and produce a piece of mRNA which has a complementary sequence of organic bases to that coding strand. So there's the mRNA in blue. Now, this will have the same sequence of organic bases as the non-coding strand of the DNA here, since they're both complementary to the coding strand. Obviously, you'll have to take account of the fact that the RNA will have uracil replaced for thymine, which will be in the DNA. Okay, but apart from that, it's got the same sequence of organic bases. So here comes our mRNA. Now this mRNA is not yet ready to be put through a ribosome, okay? Firstly, you have to modify it. So this mRNA here is what's known as pre-mRNA. Okay, so basically there are portions of this pre-mRNA which were never ever meant to actually be translated into protein. So they weren't meant to be read by a ribosome and have a ribosome match up amino acids to the codons within it. That was never meant to happen. Okay, so basically we can divide this pre-mRNA up into portions which are actually supposed to be translated. Okay, so portions that you're actually supposed to use, which I'm now boxing. Okay, and these portions that you're actually supposed to use here, these are known as exons. And then the portions which are in between the exons, which are not supposed to actually be used, these are called the introns. Now, before we therefore actually send something to the ribosome to be translated, what we need to do is cut out these introns and glue the exons back together. Now, basically, the way that you do this is not utterly set in stone. Okay, so what's going to happen is you're going to cut certain portions of the pre-mRNA out, and then you're going to glue other portions back together. And basically, the final product that you then produce will be much shorter, and you can hopefully understand how you could end up with different pieces of mRNA overall being produced from the same piece of pre-mRNA. Okay, so this process of cutting out certain portions known as introns and then gluing back exons together is known as splicing. And basically there's not just one way of doing it. So for instance, to give an example, a simple example would be there might be some exons which are optional basically, i.e. you can include them if you want, but you don't have to. If you include them, you'll get a slightly longer protein. If you don't include them, you'll get a shorter protein. Okay, so basically you can end up then with different mRNAs being produced from this same piece of pre-mRNA. Okay, and these different pieces of mRNA can then go through a ribosome and produce different proteins. And the different proteins that you then produce from these different pieces of mRNA, which all have the same piece of pre-mRNA as their forerunner, basically, are then known as the different splice variants of the protein. So you'll get different splice variants of the protein, basically. Okay, so this is what can happen for these genes that code for ASIC subunits. So for instance, if we look at ASIC1, it has two separate splice variants known as ASIC1A and ASIC1B. If we look at ASIC2, it has two splice variants as well, known as ASIC2A and ASIC2B. 3 is even luckier, it gets 3 different splice variants which are known. ASIC3A, ASIC3B, and ASIC3C, and 4 is the unlucky one, it doesn't get any splice variants, it's only got one known subunit that it can actually produce. So that overall gives us 1, 2, 3, 4, 5, 6, 7, 8 different subunits which we can use to produce an acid-sensing ion channel, an ASIC. Okay, so basically this then leaves us with the question of, we've got all these 8 different subunits of ASICs which we can use to make a third of an acid-sensing ion channel. How do we then join them together? Okay, so there are 2 possibilities here. Either you make homotrimers, okay, and what this means is, homo means the same, okay, so homotrimer means that all 3 of the subunits that make up the trimer are exactly the same, basically. So you use the same one of these 8 subunits in all 3 slots, basically, and that then is called a homotrimer. Now, the homotrimers do exist. We know that many homotrimers of these different ASIC subunits do exist. Okay. In addition, we also know that heterotrimers exist. Now, in heterotrimers, what you have is you don't all have all the same subunits in all 3 slots. Okay, so hetero means different. So the subunits which you use to make the trimeric ion channel are different, basically, and those are called heterotrimers. Okay, and those also exist. So there are different ways of combining these subunits together to make an entire ASIC ion channel, and they're not necessarily all homotrimers. You can mix and match a little bit to make heterotrimers. Okay, right. So let's now discuss the fun bit, which is what they are actually activated by and what they're going to do to the electrical potential difference across the cell membrane. So basically, their ligand is protons. Okay, so a very small little ligand. Okay, a fundamental particle, in fact. Okay, so protons, which are in free solution, can then come and bind to the subunits of an acid-sensing ion channel, and what they will trigger is the acid-sensing ion channel, which is usually closed, will then open when the protons bind. Now, of course, you have multiple subunits, so multiple protons will bind to the acid-sensing ion channel in order to cause the conformational change, which will allow the entire thing to open. Okay, right. So let's now discuss when it opens due to the presence of protons in the extracellular fluid. And I should have said that. The protons need to be in the extracellular fluid, not in the intracellular fluid. The ligand, as usual, binds from the extracellular side rather than the intracellular side. Okay, so they are activated by extracellular acidosis, basically, which means a too low pH within the extracellular fluid. Because remember, physiological pH is usually around 7.5. To activate these things, you need to go beyond 6.5, basically. Okay, and different ASICs produced out of different subunits will have different pHs at which they actually activate. Now, the highest pH that one has been found to activate at is 6.5. So the threshold for the absolute threshold for the activation of them is that you need to go beyond 6.5. You need to go lower than 6.5. So you need an acidosis, basically. Okay, right. So what's going to happen then to the electrical potential difference of the cell membrane when we open these channels? Well, they are permeable to sodium ions, okay? They're permeable also to potassium ions, although they're about 10 times more permeable to sodium ions than they are to potassium ions. And they will, at a push, let calcium ions come through, although their calcium permeability is actually pretty low, so it's pretty pathetic, really. So the main thing that they are permeable to is sodium ions. Okay, so let's discuss what's going to happen then to the electrical potential difference when you open this acid-sensing ion channel. So let's firstly discuss what the electrical potential difference across the cell membrane actually means, because I want to make sure that we're all on the same page with understanding this. Okay, so electrical potential is this concept from physics, okay? And basically what it is a mathematical model to understand reality, okay? So we're discussing electrical potential, okay? And this is the model, basically. What you do is you go to every point in three-dimensional space, so I'll draw a three-dimensional graph kind of thing. You go to every point, three-dimensional space, and to that point you ascribe a real number, okay? So some number within the real line. And that can be a positive real number, it can be 0, it can be a negative real number. It can be an integer, it can be a rational, it can be an irrational, it can be a transcendental. Whatever you like. As long as it's a real number, you can ascribe that number to the point. Okay, so every point in three-dimensional space is given this number, which is called its electrical potential. Okay, now, basically, the units of electrical potential are volts, okay? So electrical potential is measured in volts. So in principle, I can have a little man here with a clever machine which can measure electrical potential, and he can go to any point in three-dimensional space and he can measure the electrical potential, and it will give him a readout in volts of that space, at least in principle. Okay, so now let's discuss the cell. So basically, all of the points in the cytoplasm of the cell, at least to a good approximation, will be in electrical equilibrium with one another. okay? Which means that the electrical potential of all of the points that are within the cytoplasm of the cell is the same. So there is one number all over the place. If this little man took his machine and went all around the cell, he would get the same answer everywhere, basically. So we'll call that the electrical potential, big E, intracellularly, okay? So whatever number shows on his machine when he walks around this cytoplasm, that's what we'll call the electrical potential intracellularly. Meanwhile, all of the points of the extracellular fluid will also be in electrical equilibrium with one another, and therefore wherever he moves around in the extracellular fluid, the electrical potential will be the same. Okay, so we'll call that the electrical potential extracellularly, big E for electrical potential, and then subscripted E for extracellularly. Okay, now these two numbers are not necessarily the same thing. Okay, so to transfer that over onto this picture, we've got some electrical potential extracellularly, and we've got a different electrical potential intracellularly. Now, what is meant by the electrical potential difference across the cell membrane? Okay, well, the electrical potential difference across the cell membrane is also called the voltage across the cell membrane, and strictly speaking, what people should say is the electrical potential difference, or the voltage, from extracellular to intracellular, okay? And people often drop the from extracellular to intracellular, but strictly speaking, they should orient it. They should tell you which direction they're going in. Usually, it is assumed that you understand that we mean from extracellular to intracellular, and therefore people just say the electrical potential across the cell membrane. Sorry, the electrical potential difference across the cell membrane. Okay, right. Now, what it means is, if you had the little man standing in the extracellular fluid with his machine and he was measuring this number, okay, and he then moved from the extracellular fluid into the intracellular fluid, the number on his screen would change. It would change to this number. What we are now asking is how much does the number on his screen change when he moves from there to there? Okay, so that it's basically the difference in electrical potential, the change in electrical potential he will feel if he moves from extracellular to intracellular. So, what's the answer to this in terms of the electrical potential intracellularly and the electrical potential extracellularly? Well, basically we're going from extracellular to intracellular, so we want the new electrical potential, the new number on his screen, which is the electrical potential intracellularly, and we want to subtract off the old number on his screen, which is the electrical potential extracellularly. Okay, and that will tell us how much the number has changed by moving from extracellular to intracellular. Okay, so that's what is meant when people say the electrical potential difference across the cell membrane. And usually it's kept at around negative 65 millivolts. Now, of course, the electrical potential difference will have the same units as electrical potential because we're just taking one electrical potential from another, so it's going to have the same units, basically. Okay, so what does this mean? This means that if you move from the extracellular to the intracellular compartment, the change in electrical potential that you will feel is negative 65 millivolts, which means that you go down in electrical potential by 65 millivolts. So the electrical potential intracellularly is lower than the electrical potential extracellularly, and it's lower by 65 millivolts is what that means. Okay, right. So, now, we are going to open this acid-sensing ion channel when we get an extracellular acidosis. Okay, so when proton concentration is too high extracellularly, protons will start binding to the acid-sensitive ion channel subunits, and the acid-sensing ion channel, the ASIC, will open. Okay, now the main ion it's permeable to is sodium. We now need to work out which direction is sodium going to move. To do this, we need to both consider the electrical gradient and the concentration gradient. So let me give you some data on the concentration gradient of sodium across the cell membrane. Basically, extracellularly, you have a concentration of sodium of around 145 millimolar, whilst intracellularly you have a concentration of sodium of around 12 millimolar. So you have around a 12-fold concentration gradient. So that favours the movement of sodium into the cell simply because a sodium ion on the extracellular aspect is 12 times more likely to hit the channel from the outside and go in than is a sodium ion on the inside to hit the channel from the inside and go out. Okay, so you're just 12 times more likely to get an ion going this way than this way. Okay, so that favours the movement of sodium ions in. In addition, we have to consider the electrical gradient, okay, because these are charged particles. Sodium cations are moving through this channel. Okay, so at the moment the intracellular electrical potential is 65 millivolts lower than the extracellular electrical potential. Positively charged ions want to be where the electrical potential is lower, which at the moment is the intracellular the intracellular compartment. So, basically, both the concentration gradient and the electrical gradient agree. They both say move sodium in, basically. So indeed, when you open this channel, you're going to get a net movement of sodium ions coming into the cell. Okay, and of course you won't just be opening one of these, you'll be opening many of them. Okay, so you're getting this current of sodium into the cell. Okay, because simply a current just means a movement of charge. We have a movement of charge here. We're moving sodium ions into the cell, so that is a current, a sodium current into the cell. Okay, so what we now want to discuss is what the effect of this sodium current on the electrical potential difference across the cell membrane is going to be. Okay, so basically, when you dump positive charge into an area, that raises the electrical potential of that area. We are dumping sodium ions into the intracellular compartment, so we are going to be raising the electrical potential of the intracellular compartment. Okay, so let me repeat that, because it's worth repeating. Sodium ions have a positive charge. Where you have positive charge, the electrical potential of that area will be higher. We are moving positive charge into the intracellular compartment, therefore the electrical potential of the intracellular compartment is going to go up. Okay? Meanwhile, we are removing positive charge from the extracellular compartment. We didn't just get these sodium ions out of thin air. We had to take them from somewhere. We took them from the extracellular compartment and dumped them into the intracellular compartment. When we remove them from the extracellular compartment, we are going to lower the electrical potential of the extracellular compartment, because whilst they were there, they were boosting the electrical potential of the extracellular compartment. So we're going to lower the electrical potential extracellularly and raise the electrical potential intracellularly. Now, what effect will that have on the electrical potential difference across the cell membrane? Well, originally the electrical potential difference across the cell membrane was negative 65 millivolts, which meant that the electrical potential intracellularly was 65 millivolts lower than the electrical potential extracellularly. But we're now moving this one up and this one down. Okay, so they're moving in towards one another, basically. So the amount that you're going to have to step down when you move from extracellular to intracellular is going to get less, basically. So this number is going to become less negative, basically. The step down in electrical potential that you undergo when you go from extracellular to intracellular is going to become less, basically. Now, there is a fancy name for that. It's known as depolarisation, basically. So when you lower the negativity of the electrical potential difference across the cell membrane, it's called depolarisation of the electrical potential difference. Now, this then can trigger the activation of an action potential, basically. Okay, so an example of where you have ASICs is in peripheral nociceptors, which are going to sense extracellular acidosis, which should give rise to a pain signal. For instance, if you squeeze lemon juice into a cut, it will cause pain, and the reason is that lemon juice is quite acidic, And the protons within that lemon juice will be activating the A6 within your peripheral nociceptors, and they'll be triggering a pain response because you'll be activating the action potentials firing up those nociceptors, because the A6 will depolarize the electrical potential difference within the axon terminals of those peripheral nociceptors, which will then result in an action potential firing. Okay, right. So basically, when A6 open, they're going to cause depolarization of the electrical potential difference across the cell membrane, and that's important for their function within peripheral nociceptors. Okay, right, so that concludes this basic introduction of acid-sensitive ion channels.